I would like to introduce our final presentation this afternoon, which is titled Precision Agriculture, Development of Novel Tools for Crop Protection. And this will be delivered by Dr. Vava Gerbic. Dr. Gerbic holds a PhD in genetics from the University of Wisconsin, MSc in plant genetics from the University of Novi Sad, Serbia, and a Bachelor of Science in Plant Breeding from the University of Novi Sad, Serbia. Her interests lie in the genomics of plant-pest interaction, and her studies currently focus on RNAi-based pest control for the spider mite and development of spider mite silk as a natural bio-nanomaterial. The overall goal of her work is to exploit fundamental knowledge to develop novel tools for sustainable agriculture. So I'd like to welcome Dr. Gerbic, and I will hand the reins over to you.
Thank you, Cara. This is going to be a change of gears, and I would like to thank the audience for staying for this last presentation, where I would like to tell you about our efforts to develop novel tools for crop protection. This is a teamwork. I am interested in spider mite xenobiotic biology, and Mike Gerbic is an international leader in the spider mite genomics. He's one who secured funding and led the international consortium that sequenced the spider mite genome, bringing and allowing development of tools that are bringing spider mite research apart to application of approaches that are characteristic for precision health. So we are focusing on the two-spotted spider mite that is shown here at the heading of slides.
Hi,
because
Baba.
this is, yeah.
Sorry to interrupt, but I don't see your presentation on the screen. Have you?
Okay, sorry. I don't.
That's okay.
I thought that I already shared the screen. Okay,
it
yeah.
might have switched back, so.
Okay.
We'll try again. There we go. There we go.
Can you see it now? Yes. Yeah. Okay,
yeah.
Excellent. Thank you.
Thank you so much.
You didn't miss much, so that's okay. So here is the two-spotted spider mite that is a global high-risk pest. It feeds on more than 150 crops worldwide and is going to benefit from climate change. You can see that on the panel at the bottom of the slide, where geographic distribution of a spider mite is shown in red, and the pest pressure is really associated with the shade of red. And you can see that as time progresses, that certainly the part of southern Ontario is going to experience higher pest pressure, and in general in Canada there is going to be a northward expansion of its geographic distribution. So mites are already a threat to agriculture and food security, but they're going to be more so in a near future. Pesticides are one of the main tools to control spider mites in agriculture. However, mites readily develop pesticide resistance, and crop losses that occur due to pesticide resistance are estimated at approximately 1 billion across Canada, across all crops. So if you're a grower, you are likely familiar with the damage the spider mite can cause. However, to just bring everybody to the same plane, here I'm showing the inside of a tomato growing greenhouse, and on your left is a healthy tomato crop. On the right is crop that has been infested by a spider mite. So the grower, upon detection of a spider mite population, supplied the pesticide. However, that particular mite population was resistant to it, and the pesticide application was not effective. The producer applied the second pesticide, third pesticide, fourth, and fifth, none of which were effective. Finally, the sixth pesticide that was being applied was effective, but as you can see, by that time, spider mite caused sufficient damage on tomato plants the grower had to abandon that part of a greenhouse. So the question that we want to address with our work is the following. That the grower could have used the 6th pesticide first and avoid damage that occurred to his crop. So with the help of Nikki, Cara, and Tom, we have collected in the fall of 2018, we collected 19 spider mite populations that were collected mostly from tomato producing greenhouses. However, there was also eggplant, cucumber, and pepper producing greenhouse. And we have genotyped, determined alleles in targets of the most frequently used pesticides. Resistant alleles in these targets are here depicted in red, and you can see that the resistant alleles that are associated with resistance, mite resistance to pesticide, are very frequent. Not only that they are found, some of them are found ubiquitously in almost all mite populations. You can also see that the individual mite populations are carrying resistant alleles against multiple pesticides, which is consistent with the experience from the production. So really faced with this problem of multi-resistance in spider mite populations, we have organized a team that brings experts from academia, industry, and government in order to, based on genomic resources that we have created, to develop tools for spider mite pest management. And we are taking a two-pronged approach where we are developing a spider mite pesticide resistance management tool, and in parallel we are identifying novel spider mite pesticide targets that would enable development of new pesticides. In reality, I would like to use this presentation as an opportunity to explain you the strategy that we are taking and to give you a progress report on where we are and how we can work together in order to advance this work. So let me first start with explanation of our work on the development of spider mite pesticide resistance management tool. I will use abamectin. So AVID is abamectin is an active compound of a pesticide AVID, here on this slide, you can see the results of a bioassay where we applied abamectin at the concentration of LC90, and we were recording mortality of these populations collected from greenhouses. They are labeled in numbers, as well as laboratory strains that are labeled with letters. And if we take arbitrarily 30% of mortality upon pesticide application as a threshold of separating resistant versus susceptible mite populations, you can see that there is a substantial number of greenhouse populations that upon application of abamectin AVID are having a very low percentage of mortality and are therefore resistant. So with the background of this information of the presence of the resistance, we are now trying to develop tools that will enable development of pesticide resistance management tool. So the idea here is that we would be receiving samples from growers and we would be able to provide information, let's say, to a grower from the beginning of this presentation that he should be using a pesticide number 6 as the likely one to be efficient against that particular mite population, and that he should be avoiding pesticides 1 through 5. In order to develop this pesticide resistance management tool, we have to prepare 2 parts of it. One is we have to develop a genotyping platform for detection of spider mite pesticide resistant states. And I would illustrate that with this schematic that was being borrowed from depiction of antibiotic resistance in bacteria. And that one is very applicable because superbugs that are roaming our hospitals are very much equivalent to multi-resistant spider mites that are present in greenhouses. So let me illustrate what are these pesticide-resistant states that we would expect. If we are to change the meaning of this symbol so that it now depicts pesticide instead of antibiotic, then the mode of action of pesticide is to bind to a target protein. Upon the binding, the protein is no longer having activity. If that protein is performing essential biological function, then the cell and the organism is going to die. So in order to, we have the pesticide resistant state where in the presence of a pesticide, the organism lives. And let's see how that can happen. So either there is a decreased uptake or very effective pumping out of a pesticide so that there is no accumulation in the cell, or there are two classes of mutant states that would lead to pesticide resistance. One is modification of a target, either through overproduction of a target protein, so that even in the presence of a pesticide, there would be some proteins that are not being affected, unbound by it, or that there is modification of protein sequence of a target so that the pesticide can no longer bind to it. The other mechanism of pesticide resistance invokes involvement of inactivating enzymes that have capability to either cleave or to modify the pesticide so that it's no longer available or no longer capable of binding to the protein, the target protein. So in the case of abamectin, the target site is, the target protein is glutamate-gated chloride channel. And there is a plethora of mutations that have been characterized that are leading to modification of a protein and preventing a binding of a pesticide to a chloride channel, as well as there is identification of a handful of enzymes, detoxification genes encoding enzymes that can be involved in modification of a pesticide. So with that information in hand, we are now determining a spider mite pesticide resistant state, and we end up with this. So here you can see that this is rather complex slide purposely. So here is a list of mite populations. Here is their resistance status. The red, they're resistant. The blue, they're susceptible. Here is lack or presence of mutation in the target protein. And here are levels of expression of these detoxification genes. Again, red corresponds to overexpression and blue normal expression or even downregulation. But obviously, this is type of information that is too complex for us, and particularly for IPM specialists to make a decision. Well, okay, so is that mite population resistant or susceptible to this particular pesticide? And thus, we have to simplify this this information, and that really leads us to a need to develop a decision support software system, which is going to convert genotyping information into IPM decision support. So that is going to simplify and is going to have a very definite recommendation on which pesticide, what is the status of pesticide resistant of that particular mite population to a pesticide that is under study. So, and you can see here, we are using modeling to integrate genetic information into a single value that is referred as resistant index. And here on this graph, you can see the relationship between the mite mortality upon application of abamectin, and the value of resistant index. Resistant index has a value between 0 and 1, and we arbitrarily chose a value of 0.5, where any value in resistant index that is below 0.5 would be deemed any mite population that upon genotyping would end up with the resistant index value that is below 0.5 would be deemed susceptible, and anyone that has a great value that is greater than 0.5 would be deemed resistant. And you can see this is a coefficient line that is quite well in providing correlation between the observed mortality with resistant index. And you can see that out of 27 populations that we tested, we have only 2 that were predicted by resistant index to be susceptible, that are actually resistant because their mortality falls below 50%, and it's approximately 30 to 40%. So at present, this IPM decision support for abamectin can provide the diagnostics for mite resistance to abamectin with the accuracy of approximately 80 to 90% of cases. So, well, we had 27 populations that we tested so far. If we have more populations to test the model and to refine the model, we would be able to increase the accuracy of that prediction. Just on the side, so far we have developed this pesticide-resistant management tool for abamectin and toxazole. So these are active materials of avid and tetrasun. And we would continue developing these tools for the remainder of pesticides on this list, prioritizing these ones that are colored in this table. So as we are performing these experiments, here is just a side note, but of a practical importance. So here, this is the same relationship bioassay of mortality of spider mite populations upon application of abamectin. In red is one that I was already showing you, and this is one that we detected when we performed the bioassay in 2019. When we received these populations from greenhouses, we established them in the lab. We are rearing them on the bean plants and without any application of pesticides. And you can see that 6 to 9 months later, populations that used to be resistant to abamectin are now, well, they are quite susceptible. And we are seeing that with abamectin, we are seeing that with the toxazole. So in general, we are seeing a loss of pesticide resistance. Well, this is great if this is also happening in greenhouses. The IPM strategy is rotation of pesticides. So we are definitely seeing a very fast loss of pesticide resistance across pesticides that we tested. And I'm just wondering whether the lab conditions that we have, whether they truly reflect what is happening in the greenhouses, and thus we would like to have more inputs from growers as they are providing us with spider mite populations to if they can provide us a timeline of application of various pesticides, because lag of 6 months of between application of the same pesticide, I think is common in a practice, and that should lead in re-establishment of a susceptible state. However, from the practical point, that means that populations that we have now in the lab are actually susceptible to all pesticides, and we can no longer continue mapping pesticide resistance because it's just not there. And thus, I would like to appeal to growers to have spider mite populations in their greenhouses if they can collect and send these populations to us so that we can continue our work. Well, so spider mite pesticide resistance management tool is intended to extend the useful life of existing pesticides. However, it is useful as long as there is a pesticide to which a particular mite population is still susceptible to. However, we are experiencing that the number of pesticides that are available on the market is diminishing. On the other hand, we have increase in frequency of mite pesticide resistance, and that prompts us to think that we should be also putting our efforts in development of new pesticides through identification of novel spider mite pesticide targets. And in particular, we are interested in development of RNAi-based novel pesticides. So let me, explaining this strategy, let me start that with explaining how does RNAi work. RNAi is RNA interference. It is a process that is happening in our cells. It's also happening in the cells of mite, and is triggered by double-stranded RNA. So this is a configuration of RNA which is unusual for the cell, and it triggers, when it's present, it triggers a process in which this double-stranded RNA is first chopped into smaller pieces, then in interaction with some of these protein complexes, then these two strands are separated, resulting in a single-stranded short RNA fragment that is then being paired with mRNA, which is present in the cytoplasm. This messenger RNA is one that is carrying information on the protein synthesis. Recognition between the small RNA and the messenger RNA is based on complementarity, identity, the complementarity of the sequences that are present in these two molecules. Upon formation of this complex, the messenger RNA is cleaved and then degraded. So the presence of double-stranded RNA that has complementarity with messenger RNA results in prevention of protein synthesis that is encoded by the gene and by this messenger RNA. So just like a classical chemical pesticide that worked by binding of a pesticide molecule to a protein, RNAi pesticide would work by using double-stranded RNA, which is going to prevent the synthesis of a protein, and thus, if that protein carries essential function, the organism is going to die. But how would this RNAi pesticide work? The intention is to deliver double-stranded RNA to the pest. There are various ways to do it, but what is really taking popularity in the field is a sprayable RNAi. So from the grower point of view, application of a sprayable RNAi biopesticide will be the same as application of a classical chemical pesticide. So there is going to be formulation that is now, instead of a chemical, would carry double-stranded RNA or equivalent molecule that has the ability to trigger RNAi response. And upon spraying on the canopy, the pest would feed and would ingest the double-stranded RNA that would then, within the body of the pest, trigger RNAi and would result in the death of the organism. Stability of double-stranded RNA is a major issue here. There are external stimuli, factors that would affect the stability of double-stranded RNA, but these are approaches that chemical companies and companies that are developing this technology are solving by coming up with innovative formulations that are going to stabilize it. However, internal factors that are affecting stability of double-stranded RNAs that include presence of nucleases and gut pH are something that is not easy to change. And thus, for some insects, the presence of nucleases and the pH, which is extremely acid or basic, are a real stumbling block in development of this technology. Luckily for us, this RNAi is very efficient in spider mites. So here I'm showing you data where we are providing a proof of concept that sprayable RNAi can work as a biopesticide against mites. So here we tested on this graph, we are showing mite fecundity as well as mite survivalship upon mite exposure to double-stranded RNAs that are targeting different mRNAs. And you can see that there are some double-stranded RNAs that are, upon application, are resulting in 100% mortality of mites within 10 days. On the good side also is that some of these double-stranded RNAs are also resulting in diminishing fecundity of mites. So there are some of these targets and double-stranded RNAs that are very effective in reducing mite population. So in summary, we are developing two approaches to help growers combat spider mite infestation and to help with pesticide resistance management of mites. So one is development of pesticide resistance management tool. This would provide a short and medium solution to the problem. We anticipate that we will be ready. We are already ready to do diagnostics for abamectin and etoxazole, avid, and tetrasan, and we are looking to expand our ability to diagnose further resistance to other pesticides. RNAi pesticides, that development would require identification of sensitive targets that would then have to be licensed to industry that would then develop novel pesticides because there are other technologies that have to be combined with the knowledge of a sensitive target in order to have a novel pesticide. I would like to thank you for your attention and to acknowledge the contribution of people in the lab. So Vlad, Hannah, and Jeremy are working on a pesticide resistance project. Nico and Samir are working on RNAi. And I would also like to thank our collaborators, Ian Scott from Agriculture Canada, Dan Lizotte from Western, Nikki, Cara, and Tom, who are helping us at this interface with greenhouse growers. And I would like again to ask for your contribution of spider mite populations. Here are our emails. Cara allowed us to put her email, but I think you can either communicate directly with either me or with Vlad, or with either Nikki, Cara, and Tom to send and to organize sampling and sending of these populations. Thank you.
Thank you, Baba. That was great. Yeah, pretty exciting stuff. So we have a couple of questions already. So one is, what type of beans had a susceptible population? I think you mentioned that early on.
Well, so we have, these are live populations that we are working with the London laboratory strain that is in the lab for the last 10, 15 years. So These are mites that are not recently pulled from production. They have been babied in the lab for too many years, and thus they lost whatever potential resistance they have lost it since and are showing a full susceptibility. It is interesting that greenhouse populations, we received some samples from organic greenhouses. Even they had genetic markers of pesticide resistance. It's really also the question, where are these populations coming from and what is their structure, genetic structure, and so on. So this is something that we're hoping we will be able to address.
Great. Okay, so another question, where can we get more information on this? Are there papers or articles?
There was Greta Chui wrote a very good article, and I saw that she was on the list of participants. Greta, thank you very much. That was very nicely written. So certainly in Greenhouse magazine, there was a very good description of these technologies that we are trying to develop. Certainly there are scientific papers that are either in press or that we have already published, especially in the realm of RNAi. We are yet to publish our data on abamectin and etoxazole. I'm hoping that this presentation and participation in this conference would trigger the interest so that through Ontario Greenhouse Vegetable Growers, OGVG, we can organize a webinar for interested growers or anyone else. Our intention is to establish a pipeline. We do have SOP how to prepare samples, to establish a pipeline and this dialogue. We certainly need mite populations from production in order to advance this research. As we are doing that, we can already start giving back some information to growers on the pesticide resistance status of these populations that we are testing for other elements as well.
Great, thank you. So another question about how long does it take for a mite population to develop resistance?
Well, so in literature and empirically, mites are developing resistance within 2 to 4 years after a new pesticide is launched. However, when you look modes of action, so mode classification of these pesticides as they're coming, that they're commercialized, they are falling within the same group. New pesticides are falling within the same group of, basically they're binding to the same protein targets that some of the previous pesticides we're binding to, because there is limitation in identifying a new target that is sensitive to either chemical binding, or here for application of double-stranded RNA is a real challenge. So there is limitation there, and then chemical industry is reusing the same target, the same complex, let's say. There is already established sensitivity of that complex with mite resistance to previous pesticide that is binding to the same complex. So yes, there is a new pesticide that is now binding to another site, but the configuration of the complex is already slightly altered due to resistance, built resistance against the first pesticide. So the key is of developing pesticides that have completely different targets. So there is such a bottleneck in terms of pesticides that are available that finding pesticides with new mode of action is a real gold mine. And actually Tetrasan, for example, where etoxazole is targeting chitin synthase, that is a new mode of action, a new target for a pesticide. No wonder etoxazole is Tetrasan is quite effective against mites, but there is a single mutation that is sufficient to bring 100% resistance in mite population to it. So really, the IPM is complex because there are so many limitations, and on the other hand, the mites have they have genetic wiring that is really making them readily developing resistance to pesticides. And this is really coming from their ability to use multiple plants as a host. So they have evolved that chemical plasticity to deal with different toxins that we are now seeing repercussions of that in the context of pesticide resistance.
Great, thank you. Okay, another question. Actually, I'm going to address a question that's not directly to you, but there was a question about whether the recording of this presentation will be available. And I will mention that all of the recordings for all of the presentations will be available, I believe, until the end of November. So that will be accessible to you. So next question for Vava. How would you suggest incorporating biocontrol with pesticides in spider mite management?
Well, Cara, I think you're actually better suited to address that question than me. But let me bring the concept of RNAi biopesticides. They are supposed to, because that type of biopesticide would be very specific and thus would not have would only have effect towards targeted organism and would not affect any other biocontrol beneficials or pollinators, which is why the industry and scientific community is now so actively working on development of that. Incorporation of biocontrol with classical chemical pesticides, okay, I would leave that to Cara to address.
Yeah, no, I think that's a great answer though, because it is a huge concern when you're applying beneficials, right? That you don't want to kill them off as well. And it's variable, but some of the miticides we use will kill everything that's there. So, yeah. But yeah, that's definitely an interesting approach with the RNAi.
Yeah, I think that this is the best quality of that potential technology, its specificity.
Yeah, that's great. Okay, another question. Are you aware of any resistance to highly refined horticultural spray oil where the mode of action is suffocation?
Yeah, I am not. And these pesticides that are exerting physical that are affecting pests physically, I think are really the best because development of the resistance against them is very hard to achieve. Yeah, I am not aware of resistance toward that.
Okay. I suppose behavioral, like avoidance or something might be a possibility, right? But
yeah.
Anyhow, okay.
No, I was actually, sorry, yeah, go ahead.
No, you go ahead, for sure.
Okay, I was actually interested in Dave's presentation earlier in this session. I mean, he was showing these residuals in a context of microorganisms, but really there is a leftover of a past of arthropods, insects, and mites that are flowering immediately upon the reintroduction of a crop. So I did not talk about that, but Greta did write about it in her article. If one can introduce rotation, which obviously is not used very frequently in modern agriculture, but if one can rotate the crop in a greenhouse, that would dramatically reduce certainly mite populations. So, I mean, people typically have a problem with the spider mites on tomato, let's say, and cucumber, but less on pepper and maybe some other crops. So if they can be rotated, I think that would actually dramatically reduce spider mite population in the greenhouses.
Mm-hmm.